Started then. Welcome, everyone. So happy that you chose to be with us today. Excuse me. For the strategies for small patient populations. This is session 2.1, and we just wanted to welcome you. My name is Pam Todd, and I am on the patient engagement team at Global Genes. And we have wonderful speakers here that I will introduce in a minute, but first let me give you a few little housekeeping things. You may have heard this before in your other sessions, but just in case it's new to you, you will be muted, but you can ask any questions you want in the chat function. And please consider this a very interactive discussion. We are going to leave plenty of time at the end for questions. So anything that you would like to know about, direct to any one of the participants or all of them, put it in the chat. And if we can't get to it today, we will definitely follow up with our speakers and get your answers. If you have any technical difficulties at any time, just go back to the lobby, click on the help desk, and there's a tech team that will help you. And in one other thing, of course, after this is your lunch break, but we're going to kind of try to keep you on track. So let me welcome our speakers: James Valentine from Hyman Phelps McNamara, Tricia Gooljarsingh from Momenta Pharmaceuticals, and Matt Might from Hugh Call Precision Medicine Institute at the University of Alabama at Birmingham and the NGLY1 Foundation. So our process here is I'm going to turn it over to James, and all of them will share a little more background about themselves during their presentation, and they're going to hand it off one to the other. So James, you're on deck first.
Great. Thank you so much, Pam, for setting that up and for the introduction. I'm James Valentine. I am an attorney at a boutique food and drug law firm in Washington, D.C., Hyman Phelps and McNamara. So we're focused on everything that's related to FDA regulatory issues. And I've been there for the last 6 years, where I've been focusing in new drug and biologic development and approval issues, largely focusing in rare disease product development. I came to the firm actually from the Food and Drug Administration, where I was not an attorney, but actually in a unique position as a patient liaison. So my job within FDA was to engage and bring in the patient voice into agency decision making. So I was working with patients and caregivers and advocacy organizations, both on medical product decision making as well as different policy making. And as part of that, got to help create and launch the patient-focused drug development initiative. So our session today is on strategies for small patient populations. I can guarantee that some of the topics the three of us will be talking about today are things you've likely heard mentioned before, but what we wanted to do, and particularly what I'll be doing, is breaking down some of the reasons that we even need strategies for small patient populations. Some might be more obvious than others on the unique challenges that we face in rare disease drug development. And then each of us will be providing different perspectives on some of those strategies to help overcome those different challenges. But to maybe set this up a little bit more, we are in an era of real innovation in rare disease product development. These are just a very, very few of the many different innovations going on. But with me and my regulatory hat on, focused on how do we navigate to get that FDA, get drugs developed as efficiently as possible, and then ultimately get that FDA approval. I'm looking at things like benchmarks that in 2018, there were more orphan drugs approved than for more common prevalent diseases. That was the first time ever. We're seeing, real advancements in the different types of therapies that we have. We had in 2019, the first systemically administered gene therapy approved. That was the Zolgensma for spinal muscular atrophy. And in my mind, I was thinking, what really has been driving innovation? Both the greater numbers of successful orphan products being able to get approved, as well as more advanced therapies for rare diseases. And to me, there's a number of key trends I've seen. One is heightened patient engagement. Just one example of this is there's an opportunity for patient organizations to bring their patient community to FDA and to drug developers to really help learn from patients as experts in their disease. There's been 33 these externally led meetings since the program was launched in November of 2015. So there's a greater understanding of the patient experience and patients' preferences around treatments, which helps in a lot of ways. I've also seen, as I mentioned, this kind of emergence of targeted therapeutic approaches. So we're able to not just treat kind of broad approaches to trying to treat an underlying disease, but we can actually target the direct cause of a disease much earlier down, upstream to try to halt the whole cascade, of the underlying disease pathophysiology. And so, antisense oligonucleotides, gene therapies, just some examples of that. And, there have been additional incentives in recent years for rare diseases. Of course, we've had the traditional incentives for orphan products since 1983 with the Orphan Drug Act, but we have had the Rare Pediatric Disease Priority Review Voucher Program, although currently slotted to sunset, has provided additional financial incentive to develop and get drugs approved for rare pediatric diseases. And I would say that these trends, while there is this, these more recent trends, there has been a long history of FDA focusing and trying to think creatively about how development programs should be structured when you have small populations in a rare disease, and how they apply their own rules. And so in an analysis that I co-authored, and it was an update to an analysis for my colleague Frank Sasanowski, we see that over the decades, FDA has actually been very consistently flexible, applying some type of flexibility, whether it's some kind of special program that they have on their books, like for example, accelerated approval, or one of their single study approval authorities that allow them to approve on a product with just one pivotal study, or something that's kind of not on the books, their discretion. And about 2 out of every 3 approvals over time have utilized some type of flexibility. But even given these positive trends, we still have a lot of work to do. I'm sure this is no surprise to any of you. So we still have well-established challenges in rare disease product development. I'm going to go over those in just a moment, because I think that's a critical background for this discussion. And as many of you probably know, of the 7,000 known rare diseases, about 95% of them have no approved treatment. And for those that do have approved treatments, many are far short from a cure, fall far short from being a cure. So navigating the unique issues really does require its own set of expertise. And in this quote from FDA's former commissioner, Scott Gottlieb, he says it well, in many cases, developing a treatment for a rare disease can be especially hard and present unique challenges. Each success is the end of a long uphill climb. It requires a concerted effort of stakeholders, including, and as he says at the end, patient advocacy groups. And so, I think that's, kind of the, in my experience, the truth. It is always a long uphill climb, and it takes everybody coming together. And so we want to talk about what are some of those ways that we can come together. But just before doing that.
Excuse me, the sound is cutting in and out. So I wonder if we could move on to one of the other panelists and then come back to you. We're trying to get it fixed from the tech side, if you don't mind.
Sure, no worries.
Sorry, I hate to interrupt you like this, but I don't want anyone to miss a minute of your presentation.
Okay.
So we're just going to hold on James's presentation for a minute, and then, excuse me, do you want to hand off?
Yes. I will stop sharing. All right. Tricia, what do you want to go ahead?
Yes, let me just try to share my screen. Can everybody see my slides?
Yeah, thank you everyone for your patience. Yes.
Okay.
We can see your slides now.
Okay, thank you. I'm sorry, it's James, but we'll get back. Actually, it's actually a good segue So first of all, I'd like to thank Global Genes for inviting me to give my perspectives on strategies for small patient populations. And I'll be focusing today on what is actually a very complex area of drug development. It's in pregnant women. So I have a case study that I'll be showing everyone on a disease or condition called hemolytic disease of the fetus and the newborn. My name?
We'll be having them in presentation mode, right, Tricia?
Sorry?
They'll need to be in presentation mode. Right now we're...
Okay, sorry, yes.
Okay, and if you wouldn't mind just telling a little more about yourself as we go too. Thank you so much.
Absolutely. So my name is Tricia Gooljarsingh. I am the Vice President and Head of Medical Affairs at Momenta Pharmaceuticals. I have about 2 decades of experience in the industry, and I actually started off my career as a research scientist at GlaxoSmithKline, and I was there for a couple of years, and then decided to switch track and go into the medical departments, and I've been in that area for since maybe about 15 years. The majority of my pharmaceutical industry experience has been in rare diseases, and during those 10 years focused on rare diseases, I've either supported patient advocacy and engagement, and now in the last couple of years, my department is directly responsible for it. So it's something that I feel very, very passionately about and realize how extremely important it is. So for those of you who don't know, Momenta Pharmaceuticals is a biotech company located in the heart of Cambridge, Massachusetts. We have a robust pipeline of novel drug candidates in rare immune-mediated diseases. Our flagship program is nipocalimab. And nipocalimab is currently being studied in 3 autoimmune alloimmune conditions: warm autoimmune hemolytic anemia, myasthenia gravis, which I won't be talking about today, and hemolytic disease of the fetus and the newborn. Now, before I go into the journey that we are having right now with HDFN, I really have to give a bit of a background on the disease condition and the therapy that we're studying, so it makes sense. So HDFN is a rare alloimmune disease. Its prevalence is quite low. It's 3 to 80 per 100,000. In the US, that is about 35 per 10,000 live births at risk for the condition. This translates, because there's a subpopulation that's very severe, to about 3,000 to 5,000 patients in total. And I just want to emphasize that an autoimmune disease, it's clearly prevalent in women of childbearing potential, so there's really need to study medications in this patient population. The disease is quite burdensome. It has a heavy burden of disease. It is caused, in its simplest terms, by the incompatibility of blood types between the mom and the fetus. Now, when this incompatibility occurs, there's something called an alloimmunization or sensitization event where the mom will develop antibodies that go on to attack the red blood cells of the fetus. Typically, the first pregnancy is not really impacted, although the antibodies are generated in that pregnancy, but it comes really towards the end of the pregnancy when you probably have the greatest chance of maternal and fetal blood mixing. It is subsequent pregnancies after that first one that's really impacted if the incompatibility of the blood types between the mom and the fetus is present. Now these pathogenic, we call them pathogenic antibodies, they cross the placental barrier via a gateway where all antibodies can, it's the only gateway really that antibodies can cross across the placenta. Now if untreated, the attacking of the red blood cells results in fetal anemia, and if untreated, it can cause substantial fetal and neonatal morbidity and mortality. So it's quite a severe condition. The standard of care is very invasive. It's intrauterine blood transfusion. So you have to go into the uterus to deliver the blood. And the risk of fetal demise, even in the population as a whole, is 1 to 4%. But it's more severe, it's 20%, which I think most of you would realize that that's quite high. The risk is 20% in the severe population. There are no currently approved FDA drugs for HDFN. Now, why does it make sense for our compound, our molecule, nipocalimab? What is the clinical rationale for it working in HDFN? Well, it works by 2 mechanisms. It blocks the pathogenic transfer of the pathogenic antibodies from crossing into the placenta, and it also lowers the overall levels of these pathogenic antibodies in the mom's circulation. As you can see here in red, that's the placenta. You see that the antibodies, good and bad, can get across the placenta and affect the baby. When you put nipocalimab, which is in the light blue here, it actually blocks that gateway, so you don't have those antibodies crossing. So the biology and the mechanism of action of our drug is fairly well understood. We believe that the time is appropriate for trials in pregnant women. The Food and Drug Administration came out with a recommendation, a guidance document here. I would encourage everybody to read that. And what it does, it really gives very good recommendations of conducting studies in pregnant women, to generate the data and understanding of how the drugs work, so that that could in turn inform clinical decision making. Now, we also believe that the timing is right to study HDFN, and an interventional study is appropriate and in a population where the medical need is quite high. And these are the reasons. I can go through them. The historical outcome by mining the data, we have shown that there's almost a 100% risk of recurrence with the subsequent pregnancies after that alloimmunization event occurs. The early onset, as I said, which is the more severe population, really results in a very high fetal and neonatal mortality. The high-risk population is identifiable, thank goodness for that. And the disease can be monitored in as a standard of care. We can identify the high-risk population by their prior obstetric history. By the presence, we can actually measure those pathogenic alloantibodies in the blood of the mom. And we can also do cell-free DNA testing on the mom, so we can tell, if that fetus is going to have that incompatible blood type. We can also do weekly monitoring by a special type of ultrasound to identify if the fetal anemia is starting to occur. We have a way to be able to assess that, and the standard of care is to do that on a weekly basis. Intrauterine blood transfusions, as I said, that's used currently. Now, it's very important when you embark on a clinical trial that there is some way, we call it a rescue therapy is the term that we use. Because if your drug is not working in the trial, you have to have a way to make sure that you can treat those patients, right? You want the safety of the patient and is of the utmost and highest priority. We also have known centers of care. It's concentrated, there's not a lot of it, but known centers of care with high level of expertise and knowledge of HDFN, which is critical because it's such a high-risk condition. Now, before I go into the study that we're actually doing now, extensive preclinical and early clinical work had to be done, and we obviously had to navigate a regulatory landscape that was probably unprecedented. So the non-clinical studies, usually when you do clinical drug development, a lot of the studies on reproduction or development toxicology there's a kind of a slide to it, right? You can do it a little bit later. You have to do it before the submission. But in this instance, we had to do all of those studies, exhaustive studies, before we get into the pregnant population. We also did a phase 1 study in healthy non-pregnant volunteers to understand initial safety, tolerability, and the proof of mechanism. Now, I will say, to get to that protocol for the study was a Herculean effort. We had to get feedback from a vast multidisciplinary group, experts in HDFN, physicians. We had a bioethicist, and in the FDA guidance document, they do recommend having a bioethicist for pregnancy studies. We had interactions with multiple regulatory agencies across all of the different health authorities in individual countries, in Europe, with EMA, with the FDA, with Health Canada, etc. And most importantly, we had interactions with patients to get their insights into the condition, to learn from them and to get their input. And that's an engagement that we are going to continue throughout the development and until when the product becomes available and beyond. So I had a choice of the slide to put here, and I chose not to show the clinical study. I wanted instead to focus on what is the clinical strategy that we used here, because we could have gone in very different ways. I mean, those people are used to hearing like randomized controlled trials, etc. That's not what we did here. We actually did an open label single arm study. What does that mean? Open label means that the people in the study know the drug they're receiving, the people who are administering the drug, the physicians know the drug that they're giving to them. It's single arm of 15, very small, 15 patients, and they all, there's no comparator group embedded within that trial. They all are receiving the drug. Now, I think on James's slide, you saw the importance of doing the natural history study. The FDA advocates for doing that in their rare guidance document. You see that. And it really builds on the scientific foundation. It helps you understand the target patient population. It kind of reinforces the clinical endpoints that you're choosing. But it also can serve as a control, quasi-control or comparator group. And that is what we chose to do. So we have 2 studies running in parallel, one interventional with the drug, one that's similar to the study, but it does not have the drug. The studies are very small, 15, but even to get those 15 patients, we had to open up the study internationally. So the study is recruiting in North America, in Europe, and in Australia. There are a lot of challenges, as you can imagine. We're learning as we go. The physicians who work with us are learning as we go. But we have, we have a fair bit of confidence in what we're doing. We did find that there are varying levels of disease awareness, knowledge, and education. Now, the sites that we're working with, they're clearly experts, and they know what they're doing. But remember, like other academic institutes, other sites have to refer patients into those sites, and there are varying levels of disease awareness and education on the condition. It's also the same for women with HDFN. If they've had an impacted pregnancy and they've gone through it, I think they're fairly well seasoned in terms of what to expect. But if it's their first impacted pregnancy, there's a lot of lack of education there that we have to try to address. And we've done this by education. We go to conferences, we talk to a lot of the thought leaders and advocacy organizations, etc., physicians, to really advance the understanding of the condition. The geographic locations of the sites is also a challenge, right? I mentioned before that the areas of expertise are concentrated, so the geographic sites are not always close to where the patients live. Some of the patients live in very rural areas. So what we had to do is we had to have, in quote unquote, a white glove approach, where the clinical research organizations we work with would not reimburse, they would actually organize the travel, ground transportation, airplane travel, the housing accommodations for the women in the study, as well as to make other kind of considerations for their family, their husband, if they have other small children that, their care would be compromised if the mom had to travel and stay at a location closer to the site. The inclusion criteria is also very, very rigorous, because the antibodies start crossing the placenta pretty early in the second trimester. So the patients have to get into the study and start receiving the medication by 14 weeks of gestational age. Now, if it was a previously impacted pregnancy, the mom kind of knows that she has to come in pretty early in the pregnancy to kind of, for her care to be managed. But if it's a first impacted pregnancy and they don't know, they don't get in that early. So you have a very, very tight turnaround when patients are referred into the study for them to be able to be treated. In fact, we've lost patients because that, the urgency and the speed hasn't been fast enough. So we're trying to raise awareness of the urgency to all the referral centers, etc., so that we could have sufficient time to evaluate the eligibility and to get to the patients into the study. The other challenge we had is, and this is when you don't have a patient community, it was fragmented. There were powerful advocates who were blogging. There were some who had Facebook, networks that they had created international in nature. But for the most part, there wasn't a dedicated HDFN and patient advocacy organization. So we really had to work very hard we actually became the founding sponsor for one called the Hope Foundation, and then to connect to all of the other advocacy and professional society organizations that we would need to help to advance awareness. The patient population is small, like all rare diseases, it's especially small here. And so we had to do clinical trial websites, branded websites, and other awareness activities on a consistent and persistent basis. And of course, with all of the challenges that we had already, then we have the COVID-19 pandemic on top of it. And so the pandemic, we allowed enrollment where it was appropriate and where accommodations could be made very safely. But as you can imagine, if the sites were very far away, the travel became a little bit of a hindrance, and some of the sites actually closed because that was the stance that they had during the pandemic. I want to kind of do a call out. This is my last slide, but really do a call out for all of the organizations that we're working with. We believe that it is going to be critical for our success and critical to educate the community and critical to get the treatment into guidelines, critical to raise awareness of the clinical benefits eventually of the drug. So as you can see here, we have the entire ecosystem that we're working with. In the circles, it's I have some representative groups in there, but it's not all of them. We have far more that falls under each of the category. So with that, I hope that this was instructive, and we're very excited to be working in a very complex area of drug development, and hopefully, as we complete the study and we have some learnings, we're very we would like to continue to share those learnings with the research and the patient organizations. Thank you.
Thank you very much, Tricia. That was fascinating, and congratulations on your accomplishments in a very challenging situation. I think we're going to switch back to James, who's has addressed his technology issues, so we can hear the rest of his presentation. If you want to switch screens to James instead of Matt, that would be very helpful. Thank you.
Sure. And if you can just stop sharing your slides, there we go. All right. I apologize for the audio glitch there, everyone. But glad to see, as I was projecting, that, I think what was just highlighted were a number of the challenges in rare disease drug development that are faced. And I think it is worth just spending a minute talking about this, since we really want to lay this foundation for everyone that's on the call, because, everything that we do in rare disease product development, not only do we just have the regular challenges in drug development that we're, trying to find the best possible therapeutic approach, and we're, trying to show, that get that statistically significant treatment effect, but we don't have the luxury of having the large, thousands and thousands of person study to try to demonstrate that. And we don't have such large populations that we can, run multiple studies, back to back to back to try to kind of refine our clinical approach, our approach to studying the disease. Because once a person has participated in a study, they usually aren't then eligible to participate in the next one. So every patient that can potentially participate is precious. And so we really want to get this right. So the pressure is much more on in rare diseases. And, as has been talked about, natural history, is often poorly understood and characterized. There isn't typically a lot of research that's done, on the clinical course of the disease. And so, there's a need to go back and see if information can be available, done kind of a retrospective look at, clinical records to try to piece this together, or to prospectively, conduct a natural history study to try to help characterize that natural history. Because as you saw from the example that Tricia presented, it can serve as an external control group, but it does so much more, and we can talk about that more later. These diseases, rare diseases, tend to be progressive, serious, life-limiting, and/or life-threatening. And, given that, only 5% have an approved therapy, there's often, little precedent to go off of. We don't know much about what has worked well, but because of that, the seriousness of the nature of these diseases and, the unmet need, we have a pressure on us to try to, be as effective and efficient as possible. So if there is a product that works, we can get it to patients as quickly as possible. this may be the most obvious, but the small populations, often restrict our study design and replication. We have to be thoughtful, about how we design a trial, everything from the duration to what the target population within the disease you're going to recruit. All of these, what outcome measures or endpoints you're going to select, because all of these have implications on how big of a study you need to enroll. And if you design a study that's too big for your patient population, you're setting yourself up for failure. There's often a lot of phenotypic or disease, the way that the disease presents is very diverse. There's a lot of heterogeneity. And so there's a lot of complexity that's added by that. The more the same everyone in a study is, the more you can, more easily you can see that difference between what is the effect of a treatment versus what is the, just natural course of the disease. But to the degree that people get better on their own or progress more slowly or quickly or, have totally different symptoms, even though they have the exact same disease, that makes it a lot harder to identify if you actually have a treatment effect. Because there hasn't usually been a lot of clinical trials, there's not been a lot of work done to develop and those measures of treatment benefit, whether that be something that's measured in the clinic, something that the patient reports, or a biomarker. So, some lab value that is measured, some direct measure of the disease, those haven't typically been evaluated. And so that lack of precedent for drug development, in other diseases, you can look and see what's been done before, what is sensitive, what populations seem to make sense, but you're usually, treading, new water in rare diseases. And then, there's ethical considerations for doing studies in children, and that's not unique to rare diseases, but because so many rare diseases are genetic and do affect children, we often face those more often perhaps than in more common diseases. So just a few ideas that I had. There's, we could probably, put together, a textbook just on different ways that patient groups can help de-risk drug development. But a couple that are kind of, to me, ones to consider initially. If you haven't already invested a lot, where would I, what are some recommendations for trying to de-risk drug development to both encourage, drug companies and academic researchers to come into the space, but also to align to make it easier for the regulators when they're evaluating those programs when they come in. And so one to me is, developing an animal model of disease. And so, different animal models can be used for different purposes. I know this did come up in the fireside chat this morning, creating a model of your disease, whether it be in a mouse or some other animal, allows us, very early on when we identify drug candidates that might help in your disease, it allows us to screen those in different ways. And, the ideal way is you can actually use the animal model to see if there's, those biomarkers or clinical efficacy are affected by that drug candidate. And that provides a lot of good proof of concept and hope that the product might work actually, in patients. Another way to de-risk drug development, and this may be the most common topic ever talked about in rare diseases, but it's because it is so important, is to establish high-quality patient registries and ultimately build on that work to establish a natural history study. Having the patients, knowing the patients and identifying them, I mean, that's not, a given in rare disease. You may actually, be just one of the few known people, and you need to go out and try to help identify, as the patient organization, other people who maybe are misdiagnosed or, undiagnosed. So finding the people is important, and that's important because, they can then participate in research, including a natural history study, which I think was covered excellently in the opening session, to really make sure that it's, directed and accessible, not just to any one academic institution or one drug developer, but can really be utilized, to rise all tides for all of the research that might be going on. It's so critical to informing clinical trial design. Just, one example being, how long does it take normally for a particular symptom to progress, to become more severe? That might be useful to know because, you would want, if you have a drug that you think might stabilize that symptom, you might want to study, design your clinical trial to run for that duration. So if it takes 6 months or 12 months, for untreated patients to progress, it might take then 6 or 12 months to be able to show that between group difference, to show that the drug is able to stabilize the disease while in the natural history, those same patients would have progressed. We can use it to develop and test our endpoints, as well as was discussed in the previous presentation, can be used as an external control, either as the single control, or even if you do have a placebo control, it can be used to supplement that and add more power to the study. The final kind of approach I wanted to mention is really to think about your own community as a deep resource. You all, your experiences with the disease, the day-to-day burdens, how well treatments are working, preferences and priorities for future treatments, what are your treatment goals? You might think that can't clinicians speak to those issues? Isn't that, but the truth is, even in diseases where there is a lot of clinical data published, it doesn't capture what happens in between those clinic visits. And so, you can use different methods, whether it's a patient experience survey, surveying your community, or doing something more like a patient-focused drug development meeting, where you bring everybody together to hear directly from your patient community. Understanding what is most burdensome to patients and what is most important to treat from patients, really helps, drug development in a lot of ways. It helps us think about what we need to focus on for when we select and develop our endpoints to measure benefit in studies. And FDA has really embraced this, and it's now, it used to be just step one when you're designing a patient-reported outcome, so an outcome that a patient directly reports, whether it's, in a daily diary or by filling out a rating scale. But now FDA has broadened that and says, even if you're measuring something in a clinic, we want to make sure that whatever it is you're measuring, maybe it's a 6-minute walk test, you're measuring how far you can walk in an amount of time for a disease that affects mobility. Is that, something that actually would be important to patients? So before we pick that endpoint, let's make sure that it is. There's so much more than we can cover today. I just wanted to point to this resource from the Clinical Trials Transformation Initiative. There's a series of recommendations and best practices information at this website that shows throughout drug development, there's many different roles that patient groups can play to help support and de-risk that drug development. And I just want to end by a final pitch of saying not only should you be doing this and working with the researchers, working with the drug companies, but you should feel empowered to engage directly with the regulators. So the FDA or the EMA in Europe, obviously, or maybe it may not be so obvious, but you need to, these are big bureaucratic agencies. And so you have to find the key divisions, the review divisions or offices within that, are the areas that regulate products, whether, and there may be multiple different ones depending on the type of product. If it's a drug versus a gene therapy versus a medical device, and really target those areas. But there's so many things you can do, and this toolkit gets ever expanded year after year. Even from the days when I was at FDA doing this work, you can invite them to attend things that you're putting on, those scientific or research forums, drug development forums. You can request a listening session, which is kind of an informal meeting to begin to educate on the patient experience. Or you can do something that's much, larger kind of in format, like those externally led PFDD meetings. I've had the pleasure of helping organize, a little over 2/3 of those to date, and they're so valuable to, put everyone in the same room or now virtually bring everyone together. So that way, the agency as well as drug developers get the benefit of hearing, broadly from the patient community about those issues. Of course, you can do surveys and patient groups. You're not limited to just bringing the patient experience. You can actually drive some of those more technical issues, whether it's kind of getting FDA buy-in on that animal model or getting feedback on a new outcome measure that you might be helping drive the development of, or feedback on the protocol for your natural history study. And FDA has a type of meeting for that called that critical path innovation meeting that can allow you to have those more technical meetings. And then, I think it's worth mentioning, you can participate in meetings with drug companies in FDA. A drug company can invite you, if you've been working with them and giving them input on their program, it can be of value for you to come and, be a resource, help answer questions FDA might pose in those meetings. So with that, thank you so much. I look forward to the Q&A session, and I'm going to hand it off to Matt.
Thank you so much, James. And we may run over a little bit, but we want you to get all your questions answered. And since lunch is next, take your time, Matt, and then we'll have questions at the end. I think you're still muted, Matt.
Yes, I was. All right. Can y'all hear me now?
Yes.
Okay. Yes, I'll be covering the strategies for small populations more from the patient side. But I do want to advise in advance or offer this warning that if there's anything in this talk that looks like medical advice, please, for God's sake, don't take it. I don't have any formal medical training, even though there's some stuff in this talk that will resemble medical advice. I spent most of my academic career actually doing computer science, even today, even though now I do academic medicine. So there's just two things I want to get across. I want to tell you about my personal story, my son's story and my journey with rare disease, and develop that into a description of what precision medicine means as a strategy for small patient populations. So for me, it really begins with my son, Bertrand. This is back when he was about 1.5 years old, at the start of what many in rare know as their diagnostic odyssey. This is back when my family was trapped on undiagnosed island. That island with a population really of just one family. And for 4 years, we were on that diagnostic odyssey trying to figure out what was causing, his array of symptoms, from developmental delay to a movement disorder, to seizures, to this strange inability to cry tears, which is leading to all sorts of stuff like corneal erosion and vision issues. We were lucky around, I think this is 2009, that Duke was starting a research trial of exome sequencing. It was a research technique back then, and they wanted to see what it could be done, what you could do with exome sequencing in the clinic for diagnosing rare patients. What we did was trio exome sequencing, where they sequenced me, they sequenced Bertrand, they sequenced his mom, and they looked for mutations that could be driving his disease. And what they found were these mutations in a gene called NGLY1. And it looked like that these had knocked out the function of the gene, and that this could plausibly explain what he was suffering from. So they got some unusual caveats with this. They said that if this is the cause of Bertrand's disease, he is in fact the first and only patient anybody's ever seen with this disorder. At the time, it wasn't that common for that to happen. Now, of course, the sequencing, we do see this all the time. We see the discovery of new diseases at a pretty regular clip, thanks to these technologies. So Bertrand was really an N of one. So if you're talking about small patient populations, that back then, that was as small as it gets. And we had to figure out what to do for him. One thing I'll point out right away was that at least getting a diagnosis allowed us to expand our family safely. So we had another child safely. Just having that genetic knowledge that enabled us to do targeted techniques for having another child. And I'll also point out that my wife and I ended up writing up a paper about this process for anybody else that's interested in what you can do when you've got a child with a rare disorder and you want to safely expand your family. So to short circuit a lot of stuff, that was 4 years to get to the diagnosis. From there, I kind of split in two directions. I jumped into the basic science around the disease, but I also knew that I had to find more patients. You can't tackle a disease as just one family. You need at least a small community to do this. So I wrote a blog post that was really designed to go viral. In fact, it did exactly that. It went viral and ranked very highly in Google search results. So we did start to identify other patients around the world. And at last count, I think we're somewhere in the 70s, 8 years later, with the number of patients that we have. That led to some more opportunities. So I did start to end up meeting up with patients around the world. It led to an article in the New Yorker describing the birth of this community. That in turn led to some opportunities to work for the Undiagnosed Diseases Network as a consultant. Finding a patient community led to a natural history study at the NIH because they were so intrigued by the science. And that led to even further opportunities. So I ended up, starting to jump into academic medicine. I ended up at the Harvard Medical School on the faculty there. And also just by combining the natural history study data with the emerging basic science picture, I was able to make some predictions about what might be therapeutic for this disorder. I reasoned that Bertrand had a likely metabolic deficiency. He was missing one particular metabolite. And then I realized you could actually buy that metabolite on Amazon.com. So I bought it for him, took a bag of it myself just to make sure it was safe. I didn't die, and so the next day I just concluded that this compound had been FDA phase 1 safety tested. And then I decided after some sort of Star Trek level ethical debates that it was time to give it to him to see what it could do. I didn't know what it would do, but about 3 days after I gave it to him, I remember going up to his bedroom, and for a child that had serious vision issues because of his lack of tears, he was actually crying, waiting for his breakfast. He was actually making liquid tears. And then that was a big moment for us because it meant that maybe there's something we can do about this disease with what we've learned. We kept charging. We did a lot of work around, why these, what might be driving this. I think we finally have somewhat of an understanding of what's doing that. So those initial small tears ended up becoming kind of an ocean of science for the disorder as a whole. A lot of people say, well, that's great. You can make your child cry, but if you're a biologist, what you really want to know is, can you make it happen for flies too? So I ended up partnering with Clement Chow at the time at the University of Utah, where I was, to start studying fly models of the disease. And sure enough, he was able to give NGLI1 deficient flies this compound, and their survival went from around 20% to somewhere around 80%. So they don't cry, flies don't cry, but it did make a significant difference on their survival. So that, we really are accessing some part of the biology here with this. I ended up co-founding a company whose whole mission was actually to serve individual patients or ultra small patient populations when they had rare ion channel driven epilepsies. So we would do targeted drug screens looking for drugs for patients with these disorders. We were doing quite well in terms of helping the patients, and that company was acquired by Q-State Biosciences back in 2018. But the general approach that we use there still lives on, is still quite valid for finding drugs for these disorders. I ended up applying for grants to do drug development for NGLI1 deficiency, and partnered with some medicinal chemists, again, back at the University of Utah. And to make a long story short, we played around with planarian worms. And there's been talk about the importance of animal models. And I think, the smaller organisms are really critical in unlocking the science around a disease cost effectively. So we use planarian worms because they have a number of advantages for observing developmental disorders like NGLI1. Using this, we actually found a drug target. We found another protein that when you modulated it, actually improved the condition of the worms. And we then did computer simulations to try to hit that target. That target is called NGAZE. And we ended up actually finding drugs that do that. So we screened 200,000 different molecules against this target. 70 of them actually worked, at least in the simulation, and 14 were FDA approved. So we took those 14 to the bench and tested them to see if they really did hit this target. And one of those actually did. That happens to be, as it turns out, Prevacid. So Prevacid is an NGAZE inhibitor, and at high enough concentrations could be therapeutic for NGLI1 deficiency. Then some more stuff happened. I'll skip rapidly through that. So I ended up going to the White House to work on the Precision Medicine Initiative. And really what precision medicine means, if you go back to Obama's definition, and that's why it's important for the rare community, is that this is our scalable approach to making sure that we can find treatments for everybody. There's a lot of competing definitions for what it is, but on a day-to-day basis, the definition that's really all about optimizing health with data. And so precision medicine is a process where you collect data around an individual patient, and this can be omics data, it can be very rich molecular data around a patient, and you use that to find the right treatment for them. So we take in the data. For a rare disease patient, we're often going to start with the genome, but every form of data is in scope here. So we'll often look longitudinally at the electronic health record data and see if we can find any clues in there that will allow us to form this sort of constrained optimization problem where we can modulate things like your treatment and your lifestyle to find the optimal health for you. That's really the goal of what we're trying to achieve with precision medicine. And in the 3 years I was at the White House, that's what I spent my time doing, was setting up the scientific infrastructure for precision medicine so that we could do this more scalably for individual patients. So one caveat about precision medicine is that oftentimes you don't jump straight to a medication. You actually end up spending a lot of time doing scientific experiments. And in my experience, most of the time, you're going to be doing scientific experiments to reveal more data to get you closer to the treatment for a patient. Ended up, 3 years in total at the White House. I won't go into any details except to say that after about a year in the Trump White House, I quit. That's about all I could take. And then ended up coming down to UAB, where I became the inaugural director of the Hugh Call Precision Medicine Institute. And the whole goal was really to just take what I'd done with Bertrand and just scale it up, make this a process that we could repeat over and over and over for individual patients. And so that's what I spent the last 3 years doing, is building a scalable process for individual patients and small patient populations. To do this, we have several methods. The primary one, of course, is drug repurposing. We want to find existing therapies that work for a novel disease. One way to do this would be drug screens. So here you build some kind of model out of the patient, and it could be their own cells. It could be a model that's transfected to look like their cells. It could be a zebrafish. It could be a fly. But the whole goal here is to get a model of the disease in a dish that you can study, so that you get a very clear signature of the disease. Once you have that signature, you can combine the model with every drug that's available and check to see if there's anything out there that actually corrects it. The other thing we've invested heavily in, which is what I'm a big proponent of, because of its cost advantage, is artificial intelligence. So we built a tool called Medicanron, and what this does is it's really combining all the logical reasoning with all the biomedical knowledge we can get our hands on. The lay description I will give you is that it's as if you took old school logic, fused it with high-speed automated reasoning to achieve superhuman-like deduction with clinical insight. That is what Medicanron is designed to do. And you can ask it very low-level molecular questions like, what's an inhibitor for X? But you can also ask it very high-level questions like, what might be a treatment for X? And so, for example, if you ask it about overactive bladder, because it has condensed all the medical literature, it can say, I've come up with 145 different suggestions for overactive bladder. If you ask it about diseases for which there's no treatment, you can still sometimes find stuff. So for Fanconi's anemia, it came up with a number of suggestions, one of which was actually xylitol. I won't go into why it suggested that, but there's a logical chain of reasoning for why xylitol could be therapeutic in a disease like that. And I actually just got an email this morning showing that we got our phase 3 funding for this. So we'll have another, hopefully 5 years to continue developing this tool in the service of patients. I'll also point out that Medicanron is open source and freely available. And so anybody who wants to use it can. If you have trouble using it or issues using it, please just email me or the Institute and we will help you out. We've embodied this as a research consultation service. So we've got a team of analysts at the Institute now. They're all undergraduates, or they start as undergraduates, and they interact with individual patients or small populations and try to figure out what their next step is in their therapeutic odyssey, in terms of finding a treatment. They do follow a step-by-step process that we call the algorithm for precision medicine. I won't show you the algorithm in its entirety here, but I'll show you the center of it. The center of the algorithm is this branch in the middle where you've got your hands on the molecular mechanism behind the disease, and now you're trying to figure out, how do I undo the effect of that? And so usually for a rare disease, you get this from the genetics report typically, and then we have to figure out, okay, what does that mutation do to this patient? Is this gene now overactive? Is it underactive? Is it missing? Or has it become toxic as a consequence of the mutation? And that pushes us towards a set of strategies for finding drugs for those patients. I'll give you a couple examples of the kinds of things we've done. We dealt with a disorder called, well, we didn't even know what it was at the time because it hadn't been discovered yet, but ended up being a disorder driven by a mutation in MAPK8IP3. It leads to a haploinsufficiency in this gene. And Medicanron was able to predict that vitamin A would strongly upregulate this particular target. So after about 6 months on a special blend of I think it's cod liver oil for high vitamin A, this patient that had reached out initially at 5 years old couldn't even stand, was now actually starting to take it their very first steps. And so mom sent us this little video here, and you can see him taking his very first steps. With a lot. I also want to point out that there's a lot of physical therapy that went into this. It's not just cod liver oil. So a lot of credit to mom here for really pushing her child to the limits of what is possible. But again, this is a child who was non-ambulatory before treatment. And then about a year later, we got a video from mom saying, he's continuing to improve quite significantly. So this is him, I think back in January. So he's made significant improvements now in the 18 months that he's been on this high dose vitamin A regimen to upregulate the missing gene, or the insufficient gene. So this is this is one of our oldest patients for the Institute. So we've got sort of one of our best senses of what the progress can be here. But we've got a number of stories now. So we've been, you may have seen the diagnosis series from the column and New York Times and Netflix. Well, we engaged on one of those stories. And actually there was an update on it this past Monday. So if you go to Lisa Sanders' column this past Monday, it talks about how we identified a kind of amphetamine for this novel disorder that's a gain of function in a potassium channel. We've shut down our patient-facing operations during COVID-19 and focused entirely on repurposing for COVID-19. Just to give you a quick insight, all we've done is we've taken our process that we use for rare patients every single day and turned it on to it to COVID. And there's really, it was perfectly suited for this. So we ended up pulling in all the the genomic data, animal data, cell model data, and EHR data we could to find drug targets in the same way that we use a genetics report to find drug targets for patients, passed it through Medicanron to produce lists of potential drugs that treat COVID. And this actually has already moved straight into clinical trials in some cases because we proposed to the VA doing anti-androgens or estrogens as a therapy for severe COVID. And within 5 weeks, they actually approved the clinical trial. So it started back in late April. And again, using all the same stuff that we use for individual patient populations, just repurposed for COVID for the moment. And I said last night, another 9 trial proposals to the VA for what we've identified so far. Bertrand's benefited from this technology as well, just to close the loop on his story. Medicanron predicted that broccoli, really a component of broccoli like sulforaphane, would be highly beneficial for him because it strongly upregulates one of the things that helps compensate for one of his the deficiencies. And today he's a very happy kiddo. I don't want to say it's not a severe disorder, but his vision's perfect. He has no clinical seizures whatsoever. And day to day, he's a very happy kiddo. So he's learning to use his talker. He's getting quite good at that, as a matter of fact. And even to the point where he can ask for things like pet fish, which he now has in his room back home. The latest science has moved very quickly. We have an amazing partnership with NCATS and Retrophin. I wish I could spend an hour talking about just like the amazing stuff that this has done, and something I would encourage every patient community to do is to look at the resources available in partnership with NCATS and really forward-looking companies like Retrophin that are willing to partner with foundations and really dig into the science of drug development around these diseases. Shout out to Ethan Perlstein, because his company Perlara worked on NGLY1, and I guess it was about 1.5 years ago, came up with a number of potential drug hits for the disorder. I won't go into details about what those are, how we found them. All I'll say is that we were able to roll that into a clinical trial that was funded by NIH under effort that I'm doing with Ava Mareva. So again, NIH is a fantastic resource here for partnership and really pushing the science. And Clement Chow has continued to move forward and also identify additional drug targets that we could try to hit for NGLY1 deficiency. So I think the take home here is that precision medicine really means that there is hope for all patients. That no matter how small the patient population, if you can harness the drive and the passion of the patients, we are creating the conduits to funnel that drive and that passion, that motivation into meaningful science. And I'm privileged that I get to have a day job now where I get to watch that all day, every day. So I know we're about out of time, but I can hang on a little bit longer if there's some questions. So thank you.
That would be so great. Thank you so much, Tricia and Matt, and thank you, James. It's been a wonderful presentation. We have a few questions. Let me preface it by saying we've been trying to answer them in the chat. So James has shared a city link from his presentation. We'll request the presentations if you're willing to share them. We can definitely post those sometime in the future. And there was a question about best practices white paper or information easy to understand for advocates for developing a patient registry. One thing that I would highly recommend is downloading the Data DIY workbooks and all of the events. There were 4 full-day events. You can get just unlimited information. It starts with very basic and goes on up to how to create a patient registry, informed consent, on the natural history studies and collaborative research networks. So that's a great resource. RADAR, which is the NIH site for registries. It also has wonderful information, but is there anything else that you panelists would recommend as far as learning how to create a registry?
I'll be honest, I still use a Google Sheet.
Done an awful lot with the Google Sheet. Anyone else have recommendations?
No, I think the two you mentioned are great ones, the Global Genes Toolkit and the RADAR's website, for sure.
Okay, great. So let's see. One of the other questions we've had is, when you have a really small patient population, of course, it's going to be important to find the patients, as many as possible, and keep them engaged in registries or whatever other activities you have. Do you have any suggestions related to that?
I mean, from my perspective, you know, we have a patient mailing list. Some people use Facebook groups. So social media is a powerful way of keeping patients engaged and connected. And I think, you know, it almost happens by default these days, given that, you know, social media is so ubiquitous. It just, you know, people will form these groups. And so as long as you've got responsible moderators, you can have a thriving online patient community.
Mm-hmm.
Yeah, some of the things that we're contemplating is like for the HDFN, the model that I showed, we have like you can do patient websites. And that's another way where you can engage them with the education. But also something that I feel very passionate about is like when the clinical trial results and that sort of thing is completed, we really cater to a physician-based audience, right? So all of the peer-reviewed communication, it goes out into publications and that sort of thing. And I think the industry and as a whole hasn't been really good about, like, as soon as those trials are done, really trying to get those information out to the patients. So I think that's something that we could do better at. And certainly, you know, if, you know, they contribute to the trial, it's good to know what the outcome of that is, right? So.
Mm-hmm. Great, thank you very much. I guess Matt just dropped the link to his Dropbox with the slides. That's good to know, Matt. You're really on the job there. Speaking of multitasking. Do you have any thoughts on the FDA and other regulatory bodies, how they're shifting to further embrace small patient population studies? And also what challenges still remain in representing validated data from these studies?
Okay.
Wow. Yeah, that's we could spend a lot of time talking about that. You know, I mean, I think it is being recognized more even within FDA that, you know, reviewing data from small, you know, sample size studies is a unique skill set, you know, so that it's not enough to just know the clinical science in rare diseases, which is hard enough already. And there's a lot of educating that patient communities and industry can do, you know, but there's a lot of difficulty in interpreting data from smaller studies. You just aren't, you know, you don't have the luxury of all the things we talked about. You know, you do the best you can. You pick the best endpoints you can. You try to get as many patients enrolled as you can. But at the end of the day, you're also constantly learning about rare diseases, even as you're doing these studies to evaluate whether a drug works. And so you have to be really open-minded about that. You know, so I think, you know, across the board, there's, you know, obviously there's been a lot of rare disease drug approvals, you know, but the rare disease patient community has continued to push for additional kind of development within FDA in that area of expertise, because there's not just one division. Even though there is one division that's called the Division of Rare Diseases and Medical Genetics, you know, they're only really looking at, you know, certain inborn error products and other ultra rare diseases. Every single review division in FDA has jurisdiction over rare diseases, some with much more, you know, kind of a higher volume than others. And so you shouldn't get a different kind of, you know, venue. You know, you shouldn't be treated differently just because you happen to be, you know, in ophthalmology versus pulmonary versus neurology, for, you know, whatever it might be. And so, you know, that is something, you know, to keep in mind. And, you know, there are kind of broad approaches being taken to push for FDA to have kind of more consistency across the divisions.
Great. If you can you stay a few more minutes, everyone? Okay, a few more questions. How likely are you to get funding and attention for research when the likely solution is not a medicine, but lifestyle or diet changes?
I don't know.
Yeah.
I've never, we certainly have had some disorders where the treatment wasn't a medicine, it was actually a diet, or a natural product supplement in some cases. That does happen a fair amount. In terms of like the basic science, though, I don't think it's impacted our ability to get funding for it to date.
What excites you most at this moment in rare disease research?
I am excited, obviously, because of the engagement with the patient community, and also with all of the patient, the PROs, they call the patient, looking at measures that come directly, the symptoms and that sort of thing, getting the input on those things that really matter to them and factoring those things into the clinical endpoint. Another thing I'm seeing is that they're composite, it's called composite endpoints, in the sense that you would measure more than one thing, right? So for example, I'm using an example as a GI, a gastrointestinal disorder. You may have pain, but you may also have bloating and distension. And so instead of just doing one measure now in a clinical trial and maybe having the others placed a little bit lower on the scale of importance, they're getting like equal weight. So I think we're definitely seeing some innovation in a lot of the ways that we're trying to measure and assess that a drug works, and definitely coming with substantial patient input to do that.
Great. James or Matt, anything to add to that? What are you excited about?
For me, I'm excited about the continued scale down of the whole process to individual patients. I mean, it's every year I can see that the threshold for reaching individual patients gets lower and lower and lower, and in terms of the cost, the efficiencies, everything. And so I'm sort of drawing that line out to the future, and I see a world in 10 years where N of 1 medicine really is accessible to everybody.
Great.
And maybe I would just add, I of course think that the culture shift that's happened, where the patient is really recognized as the expert, and beyond that, patient organizations can really drive this. I don't think that was a well-tested model, before 5 years ago. So if you're a patient group, you can do as much as you want to do in terms of sponsoring research, directing the research kind of landscape in your condition. You're not just in the passenger seat, kind of putting money towards things, but letting someone else drive what gets actually done. You're not just providing patients to fill slots in clinical trials. You're actually, a partner in the process, and informing the design of those clinical trials, and helping interpret the results of them for even the regulators. So that's incredible, and it's just hard not to be excited about the emergence of the targeted therapies. Being able to see things that that would normally be extremely debilitating and life-shortening diseases, to see that be transformed by ASOs, by gene therapy. It's just totally incredible to be seeing that evolution in medicine.
Yeah. Well, I think that's a great place. Oh, did you want to say one more thing, Tricia?
Yeah, I just wanted to say one thing, because it's about what COVID-19 has kind of now unlocked, and what we're seeing in the industry is that it's this kind of burst, if you will, of telemedicine. And we're seeing a willingness from the authorities now to kind of work with us to reimagine what those clinical trials can really look like. So, before it could be, very traditional. You have sites take care of the patients and that sort of thing. But now maybe it's more remote. Maybe it's, I don't want to use the word sightless, but maybe there is oversight, and you can you care patients more on the local level, and that sort of thing. And also the way that you assess them and monitoring with all the technology and tools available. I think we're kind of seeing, we're probably going to see more of that. I mean, I hope we're going to see more of that just because COVID-19 and the pandemic is over, I hope that you know, the mindset doesn't like go back in the other direction.
Yeah, that's a wonderful point, and very, very contemporary and important today. And I love all your inspiring thoughts. And I think that's a wonderful place for us to end and go to lunch and think about the future of rare disease. So thank you all for spending time today. And you know, if anyone has any future questions, just please submit them in the chat, and we'll try to get them answered later. Thank you. Enjoy your break.
Everyone. Bye-bye.